So hi everyone. Thank you for joining the August MicroTalks. My name is Lin Zhu and I'm a coordinator for the ASM MicroTalk series. So we have MicroTalks the third Monday of every month. Due to COVID, we'll be hosting them via webinars. Victoria or Tori will be your moderator today. So Tori, take it away.
Thank you, Lynn. So again, good afternoon, everyone. I'm Tori Poplaski. I'm a fourth-year graduate student at BCM, and I'm co-mentored between Dr. Mary Estes and Dr. Robert Britton. I'm currently the Vice President of ASM and serve as co-chair of MicroTalks with Lynn, and as she said, I'll be the moderator for today. So we have two great talks for you today. Our first talk is going to be from Dr. David Marciano. Dr. Marciano received his Ph.D. from Baylor College of Medicine in the Department of Virology and Microbiology, and since completing his Ph.D., he has been a member of the Olivier Lichtarge lab in the Molecular and Humans Genetic Department, also at Baylor College of Medicine. Dr. Marciano's work focuses on bridging protein evolution, synthetic biology, and quantitative systems biology, and his lab currently utilizes cancer bioinformatic techniques in order to identify adaptive mutations in bacteria and refining CRISPR genetic engineering. Dr. Marciano has nearly a dozen publications, with his most recent one being a Nature Communications paper in 2018, which is very exciting. And we are excited to hear what he has to share with us today. So Dr. Marciano, you can take it away.
Thanks. Yeah, thanks for the opportunity. I really appreciate it. So we're mostly a computational lab. I'm kind of the head of the wet lab. I joined soon after graduating from Tim Polskill's lab and helped Olivier build up wet lab. And been working in bacterial genetics, and we've gone on to do like experimental evolution in lab strain E. coli. And so the big background of the lab is predicting the impact of mutations, looking at how amino acid changes are going to impact function. And so mostly this is done in human data sets, but I'm more of a E. coli geneticist now. And it's a kind of an obvious application to work in a system that's very much controllable, where you have control over the variables. So what I'm going to present today are selections we've done in the lab for Cipro or Colistin, and then I'll also share some data at the end where our collaborators have grown radio-resistant E. coli. So just to be sure I don't forget to acknowledge the important work of the people in the lab, and especially Chen Wang, who's done a great amount of work on this project. And I'm indebted that he has joined the lab and helped me out a great deal with this. I also want to acknowledge the instruction experiments, and my co-mentor, Christoph Herman, and our collaborators and Michael Cox and Stephen Bruckbauer. And also it's important to acknowledge our funding, which is from IARPA. This is Intelligence Advanced Research Projects. And what they want us to do is be able to detect possible agents of biological threats. And so the idea is that we could evolve bacteria in the lab and kind of learn from that in the event that we are attacked by an antibiotic-resistant agent, for instance. So just an outline of what I'm going to show today. First, I'm just going to give a very brief overview of experimental evolution in E. coli, how we approach the data that comes out of that, and then present Cipro resistance data, which is the kind of the model system where we know a lot, and then also where Chen has done a lot of the work, which is Colistin resistance data, where we don't know a heck of a lot, and so we've discovered quite a few genes that contribute to resistance to Colistin. And then finally, we're going to apply what we've learned from these two systems to the radiation resistance situation. So I was kind of surprised that when you grow an overnight culture of bacteria, the amount of mutations per genome comes out to be roughly about 10. And if you consider how many bacteria there are present in one overnight culture and the size of the genome, this means that it's pretty much guaranteed that nearly every substitution has been sampled in that overnight culture at least once. That was kind of surprising to me. And these are just back of the envelope calculations, but point is that in an overnight culture, you're going to sample a large number of mutations. And so if you apply a selection to this, you should enrich for mutations that confer a fitness advantage. And what you can do is you can take those cultures and isolate DNA, subject them to sequencing, get your reads out, and then what changes should be somehow tied to the phenotype change, or in our case, Cipro resistance, Colistin resistance, or maybe radio resistance. And so you just repeat this process independently several times, and if you see the same mutation occurring again and again, you have a pretty good feeling that, yeah, that mutation is driving the phenotype and is responsible for that change from, in this case, sensitive bacteria to resistant bacteria. And so our approach is more than just counting mutations across different replicates. What we also do is we try to predict the impact of those mutations. And we're focusing on amino acid substitutions, honestly, because that's the easiest thing to predict upon. We haven't been able to quite work out how to do this at the nucleotide level, but it's definitely possible. It's something we want to work on in the future. But the idea is that we estimate the evolutionary importance of where a mutation is occurring in a protein. And so this is done by looking at the phylogenetic tree of that protein family. And so, for instance, if it's a position in the active site, that's going to have a heck of a lot more impact on the function of that protein than a substitution that is just hanging out in solution somewhere and that's changing often along the evolutionary tree. So the other half of where the mutation is what is the mutation, right? So for instance, if you have a serine to threonine substitution, that will probably have a different impact on function than if you have a serine to tyrosine mutation. So we can look at the how often you see one substitution versus another at a particular residue in a protein family and figure out what are the chances of that substitution occurring at that position. And so that is the resulting product, which is the evolutionary action score, which should predict impact on function. So this works pretty well. There are several data sets out there that measure the impact of mutation on a protein's function, including some very, some older work on lac repressor. And so here, in this case, 100 would be the highest impact and 0 would be the lowest impact. And so you see a good correlation between mutations that are shown to have impact on function and our predictions of what we think will have impact on function. This is also true for bacterial phage enzyme, T4 lysozyme, as well as a HIV protease enzyme, and even cancer driver P53. So, you know, this is based on evolution, so it really shouldn't be specific to human data sets or bacterial data sets or even viral data sets. Yeah, if you have any questions, feel free to just post them in the chat and hopefully I will see them pop up and then I can address those as I'm going through here. So we can apply this evolutionary action score not to just single proteins, but genome-wide. And the idea is that mutations that are going to be more, that are going to impact function more, are going to have be enriched for these high impact scores. Versus if you look at all mutations across the genome, you should have much more substitutions that are predicted to be low impact. Maybe you will have some that are higher impact, but the distribution between what you see for a gene of interest and that has an impact on phenotype should be different than the entire bulk mutations you see across the genome. So the idea is that we can apply this to the antibiotic resistant data set and pick out those genes that are repeatedly getting hit by these high impact mutations and separate them out from the large bulk in the background. So as I mentioned before, the two resistances or two antibiotics we're looking at are ciprofloxacin and colistin. Ciprofloxacin is widely prescribed. It's a WHO essential medicine. Colistin, not widely prescribed, but it's a last resort. And it's gotten a bit scary because E. coli has shown up with resistance the last over 2 years ago, I believe. It's been around, but the scary part is that it's become plasma-mediated resistance. And so we're kind of running out of bullets in our armory for these multi-resistant bugs. So Cipro targets DNA topoisomerases versus colistin targets the outer membrane, although exactly how it does that is not quite clear. Maybe experts in the field have a better sense of what they think is really going on. One important thing about ciprofloxacin resistance is that the resistant mechanisms are very well studied, and there's lots of genes that have been rigorously tested and shown to contribute to resistance, including the 4 genes that make up the topos that are targeted by Cipro, as well as several regulators and genes that are involved in either transport or export of drug. Versus colistin, there's definitely 2 genes that are well described that get mutated over and over again. These are BAS-S and BAS-R, and the way they work is they act upstream of a pathway that modifies LPS. And somehow this modification of LPS makes it so that colistin either cannot bind LPS as well or cannot perturb the membrane as well. So to show you the actual, what we did in the lab, the way we set this up is we actually just have 50 ml tubes, very simple, 5 mils of M9 media each, with concentration of antibiotic that will kill 99% of the bacteria present. And then we have a backup of half that concentration of antibiotic, just so we don't have any lines that go extinct. And so we grow these for 24 hours and measure the OD. And if the OD is above a certain cutoff, then we say, okay, at this point, the MIC must have increased. And so when we do the next round, we're going to double these concentrations. And so in this way, the cultures are always exposed to a slightly higher concentration of antibiotic, but we always have a backup so that continue to grow. They won't be pushed too hard into extinction. And so when we did this, actually when I first did this with Cipro, it was with the mutator strain, and it became pretty clear that this thing was really hard to work with genetically. And so we went back and repeated the experiments either with wild type E. coli or wild type with mutagen. So the idea is that, well, honestly, for our approach, we're comparing distributions of mutations. And so we really need multiple mutations to get a good statistical reading of whether or not they vary from the background. Also, this is in many ways a more challenging system because you're getting a whole lot more mutations. In the wild type situation, you're expecting here, it could be an overnight strain of this mutator. And also, we did this both for Cipro and colistin, multiple replicates, and you get pretty much a very repeatable trajectory of increasing resistance over time. So after about 2 weeks, we didn't sequence all these, but We sequenced a total of about 150 selected cultures. And what you see is that, as expected, we have a different rate of mutations observed, or rather amino acid changes observed for under each of these mutational regimes. So wild type, we're seeing about one amino acid change per day, versus whenever we have the mutagens present, we're up to about 40, and then in the mutator, it's way up around 700 amino acid changes per day. So the question is, how is this resistance being conferred, right? So the known way that resistance occurs is through modification of LPS through these upstream mediators of LPS synthesis. But that's only the BAS-S and BAS-R part of the story. But there's probably other genes that are involved. Or maybe not. Maybe it's just these two and that's it. And we don't actually need to do these experiments. But that's what we set out to find out. And so if you look at EA profiles, this is what is going to distinguish the drivers from the background. And here is a known Cipro driver, PAR E. This is one of the targets for ciprofloxacin. And so here there's a distribution of mutations observed in the mutator strains. I'm sorry, in the wild type plus mutagen. And then in red is the mutator strain. And so you see a lot of these mutations occurring in high impact versus the dotted line is all the mutations we see in the background without selection. Compare this against a gene that has no known impact on ciprofloxacin resistance. There's very few mutations, and those mutations, they look like background, although here is just one mutation, so how can you really say one way or the other? But the point is that we should be able to use this shift in the profile to distinguish driving mutations from a background. And then here is BAS-S, one of the two known drivers. And you can see it is getting high impact mutations again and again in the three different scenarios. So right, so we only did, so far we've only done sequencing at the very end, so we don't actually have time resolved data, although that would be very interesting. And I think it's a way we could go in the future. So this is a ranking of the genes based on EA. And so the orange genes are known drivers, and so they are ranked more highly towards the top. And again, for Colistin, we have those two right at top, and then we have a whole bunch of others that may or may not be involved. And so I tested a lot of these in Cipro, and Chin tested several of these Colistin-resistant mutations. And the way we did that was we took an evolved isolate from our evolved lab strains, and we used P1 transduction with the help of Ralph and Susan's lab to revert one mutation in the background that it was found. So in this way, you don't have to worry about epistatic effects. That should be accounted for. So you're measuring the impact of that mutation in the background that it was discovered. And so the known drivers, again, are in orange. And as expected, they influence ciprofloxacin resistance. But what's interesting is in Colistin, a lot of these new genes are conferring resistance. So that is a nice, I think, demonstration how we can discover new mediators of resistance to something for a system we don't know a heck of a lot about. And so we just put together, try to put these genes in context. So a lot of these are involved in either LPS transportation or modification or synthesis, including the two known ones which act upstream. But then other ones, we really don't know how they're involved. Some of them are somehow involved in membrane metabolism, but it's not really clear what the relationship is with Colistin resistance, or if it's acting through some mediator, we're not sure. But I think this shows that this approach is successful at figuring out which genes are contributing to resistance. And so the question is, can we do this in a different system, totally different system? And so we looked at, with our collaborator, radio-resistant E. coli. And their setup is very similar to ours, where they just grow these in culture and expose to selection, dilute, and repeat. But the one difference is it's a lot harder to do this with this gamma ray source than just adding antibiotics to a tube. So they only had 4 lines that we could analyze. So this is a more challenging situation. But we went ahead and tried, and yeah, it's more challenging. So we did not do really great, I would say. So these are the known drivers in orange, and you can just count the mutations. And that actually does a really good job of separating those from the large background. We do pick up 3 out of 5, but then Chen thought, maybe we could approach this in a different way. Kind of address this sparse data issue. And instead of looking at one distribution like an RPOB versus another, maybe we could just sum the mutations over that we see across the lines, and then use this to rank each gene. And so when we do this, we get a much better result. All the known drivers come up way up the top, even better than what was seen with frequency alone. And we have some other interesting things like DNI that could be involved. So I think this is a nice example of how you come up against a challenge, and then you're forced to kind of think your way around it and then come up with something even better. So the question is, does this apply to the antibiotic resistance stuff as well? And indeed, it does hold up. At least it does not hurt the rankings that we saw before. JRB actually does come up higher than it was ranked previously. But all in all, it's not deleterious to our results from before. So overall, we can use EA to distinguish genes that are responsible for phenotype changes, and that we found that if we actually sum these EA scores rather than comparing distributions, we can get more sensitivity with fewer samples. And I feel like this story is pretty much wrapped up, but maybe we could look at adding more data, because we've tested about 24 mutations, but there are 24 genes, but there are hundreds and hundreds of genes that are mutated. So we're only looking at a fraction of what is actually mutated in the experiment. So I think I'm pretty much used up all my time, but maybe I'll ask the moderator if she feels like we can take a question or two.
Yeah, let's see what we have for questions in the chat. So someone had asked, the resistance conferred by the modifications are over the structure of LPS in the outer membrane or a biofilm.
I'm not sure about, so this is done in liquid culture. So what do you call it? Planktonic cell growth. So I don't think the biofilm is going to be influenced in these particular experiments. But certainly LPS is probably being modified based on the genes that are being mutated and are under selection and shown to confer a fitness advantage.
Great. So someone else asked, I am guessing a high rate of mutations would damage the genome in the long term. For the mutator series, does the mutation rate stabilize with time?
I don't really know the answer to that. I suspect it does. So if you look at, I mean, the mutation rate of that particular mutator is supposed to be 1,000 to 10,000-fold higher than wild type, which is basically the highest that has been observed. And we're seeing over the course of the experiment about 700 amino acid changes per day. And so that it should be way higher than that. I mean, it's already ridiculous, right? How many that is, but it should be higher than that. So I suspect that there have been some kind of anti-mutator mutations that have occurred, or maybe there's some other explanation where there's just so many mutations that you end up getting kind of degradation of the entire proteome. And so if you add any more mutations, you just fall apart.
Okay, thank you for answering both of those two great questions. Let's go ahead and move on to our second speaker, just so we can stay on time. So our second talk is going to be by Dr. Austen Terwilliger. Austen also received his Ph.D. from Baylor College of Medicine in the IMBS graduate program under the mentorship of Dr. Anthony Moresso. And during his PhD, he worked on nutritional virulence of Bacillus anthracis. And since completing his PhD, he's continued to work as a postdoc in the Moresso lab, where he works on bacterial phages and combating antibiotic resistance. In 2017, Austen became the director and founder of TAILOR, which stands for Tailored Antibacterials and Innovative Laboratories for Phage Research. TAILOR develops new technologies that facilitate the discovery of novel phages, combat multi-drug resistance, as well as other novel applications. TAILOR also aims to compile the largest characterized library of therapeutic phages for all major drug-resistant bacterial species, which is super cool. Dr. Terwilliger also has nearly 10 publications, with his most recent one being a perspective piece that was just published this past June in Phage. And with that, I'll let Austen take it away so that we can hear more about TAILOR and their exciting projects.
All right, great to see everyone here. Please just shout out if there are any technical issues with the presentation. I'd like to thank Tori and Lynn for inviting me. And as Tori said, I'm going to show you a bit of an overview of TAILOR, our mission, vision, some of the projects we're building, as well as what it takes to develop a compassionate use treatment. In this case, we're talking about patients suffering from chronic multi-drug resistant bacterial infections. And I want to start by acknowledging the reason why we got to this point, that is to say the fundamental problem that is driving these cases, and that problem is change. Bacteria are really good at changing, and through mutation, conjugation, transduction, and transformation, this is what we call the mutagenic tetrasect, the collective mutable power that bacteria wield to overcome the many obstacles that humanity puts in their place, whether they be facility and engineering controls, or vaccines, or what brings us all here this afternoon are antibiotic drugs, which don't really change as much. There's a limited amount of chemical space here with which you can make changes. And what I hope is clear, that this chemistry is not going to be able to compete with this biology here, just in the amount of change that's possible. And of course, we see this this play out, right? Who wants to invest 10 years and billions of dollars in drugs to which bacteria are going to be resistant to in a matter of months or weeks even. So as companies develop less drugs, antibiotic resistance continues to rise. And in the meantime, this mutagenic tetrasect continues to spin out and take a great toll. 2 million infections, 23,000 deaths, and $2.2 billion per year just in this country. And this is where we want to intervene. This is where we want to make a change ourselves. And we believe in using this same source of change for good in these bacteriophages, right? These viruses that specifically infect and kill bacteria. You find these phages wherever any bacteria are, which is to say everywhere really. And unlike our pharmaceutical drugs, phage are capable of evolving and changing with their host. So they can be an abundant and diverse source of antibiotic power. And we are already starting to accept personalized medicines that change, whether they are derived from your own cells or editing your genes, or prescribed for a specific form of a disease. That there's power in being able to tailor treatment for your exact illness. And much thanks and much is owed to Sabrina, or as we call her in lab, by her Twitter handle, Mother of Phage. She's defending this Thursday, I think, also something you would not want to miss. But she had started the basic science and inspired us in part to build what has turned into the TAILOR Service Center. This is a research initiative funded in part by the Baylor College of Medicine, institutional funds, and research contracts. We are a group of scientists and physicians striving to build personalized medicine for antibiotic resistant cases and serve the most vulnerable at-risk patients. I think the best way to demonstrate what we're capable of doing is to basically walk you through how a generalized compassionate use case works, and I hope you'll appreciate the collaborative culture that we are trying to build to leverage all these great resources that we have here at the Texas Medical Center. So the first step is any case is that doctors have a problem, and they are reaching out to us. They have a patient that has a chronic recurrent multi-drug resistant infection. We get contacted for many different types of cases. What is common is prosthetic joint infections, urinary tract infections, and lung infections for CF cystic fibrosis patients. And the main thing we look at first is, does this case and patient fit what is generally deemed necessary for a compassionate use case, that the risk of treatment is not greater than the disease itself, that there's really not any satisfactory alternative to look at. Again, these are multi-drug resistant infections. The doctors have pretty much used all options that they have for them at this point. So if we believe we can make a difference and help out here, the first thing is to send a clinical isolate from the patient. In developing a personalized medicine, you're going to want that pathogen that is causing that infection in that patient, so that we can start a phage hunt. I have a general timeline here. This is not an exact time, but rather what we are capable of. Oftentimes, these cases take about several months to get to the point of treatment. Finding the medicine is just one day. You, again, phage are where the bacteria are. You go to where bacteria are, which is to say sewage. Houston Public Works has been working with us and offering us access. You really just need a rope and bucket here, and It's a pretty straightforward process. We filter this sewage and you mix it with a culture of that target bacteria you are trying to kill, and you mix it in a top agar assay, and you're looking for plaques. These are zones of clearing that indicate the phage have killed these bacteria. And indeed, we have found this for any number of escape pathogens. You may note we are missing the A there. We still haven't found any Acinetobacter bomoniphages. So if any hospital would like to collaborate with us on this, we'd be very excited to do so. The bulk of this work, the engine of a lot of the basic science at TAILOR is driven by our technician. He has found the first in pterococcal and staphylococcal phages in our labs, and we keep on looking forward to great things from him. But okay, what if we can't find phages from the environment? What other options might be available to us? Well, we can. Whoop, I apologize for a second. This isn't moving. Oh, all right, great. So we can also evolve phages to find what we are looking for. This is working with Dr. Justin Clark, a postdoc in the lab, who we have developed this co-culture system where we have a host strain in blue that is susceptible to a phage, and a target strain in orange that is not susceptible to the phage at first, but we give the phage access to both of these strains. And as David was telling us from his talk, if you let bacteria go in culture, they are going to mutate and change. We basically let evolution take its course. We get descent with modification, where this host strain is going to make a progeny that might have a affinity for the target strain, be able to infect it, and if so, that is going to be favored over time in culture, and you're going to be able to evolve a phage for your purpose. We have used this method to evolve phages against strains of E. coli and Pseudomonas, and we're excited about working with this further. But anyway, if we find them in the environment or we evolve them, we are building our inventory. This is just an overview here, and we're always looking for more. If there's a strain of bacteria that you aren't fond of and wondering if we could find phages for it, please send them over to us. So in any case, when we find phages that are able to infect the host strain, the next step is purifying. All of these steps here, the arrows are overnight steps. That's why this takes several days. This is pretty standard. In the field, we're working with about several liters worth of culture, and we're scrubbing the phage in a cesium chloride gradient and dialyzing it out. We're going to get to this a little bit later, but at this point, really what we're thinking about is making sure that we have enough phage to satisfy a course of treatment, and being able to remove parts of the host, parts of the bacteria that you don't want in your phage preps. Once we have our purified phage, we can characterize them by a number of ways. This is just looking at titers and plaques here, showing that they are virulent and lytic on this host strain. You're going to want to know that your phages are stable over time. Again, we're talking about a several-week course of treatment, and often these phages are delivered in saline to the patient. So obviously, you're going to want to know, are these phages stable? in saline, and in this cocktail, I love this one. It's stable in saline out past a couple months. That isn't always the case. Our colophages often, you'll see, lose 1 to 2 logs of titer in the first month. We also want to know how virulent our phages are, and for that we do a killing assay in culture where we mix the bacterial cells and phages at different MOIs, multiplicity of infection, incubate in culture for 4 hours, and we look at how many CFUs are left. And in this case, we have four logs killing with phage compared to where there's no phage. So this is a pretty virulent cocktail here. All of this information helps us to formulate the final cocktail, what we want to give to the patient. Again, we're thinking about how virulent these phages are. We're thinking of host range if there's multiple strains that we're trying to clear inside of the patient, how stable they are, and really the most important thing is what the pharmacy is capable of. It's really important at this step to work with doctors and pharmacists to figure out what facilities the pharmacy has. Are they capable of working with a concentrated phage cocktail and making formulating single doses on site, or do we have to think about formulating single doses ourselves here at Taylor? This is a very key step, because you are not going to want to do future you aren't going to want to do more certification and testing that is needed. It will take up weeks of time, and it's costly then too at this point. So if you have a cocktail of phages that you're looking to treat the patient with, the first thing is just making sure that what you have is sterile. We use USP 71 methods. Just incubate your cocktail in rich media, grow it for 14 days, and if there's no growth, it is certified as sterile. As David had briefly talked about, and I had mentioned, we are interested in the levels of endotoxin that are present. Lipopolysaccharide is a component of gram-negative cell membranes. It causes a general pro-inflammatory response. So you want to make sure that you aren't dosing a patient with high levels of it. And we do this working with the CAG-T Center here at Baylor College of Medicine, just using a typical chromogenic assay to quantify how many endotoxin units are in your purified phage preps. We typically see about 100 to 1,000 endotoxin units per mil, and this is important because when you look at, all right, what is the titer of my phages? How many phages do I have? What volume of that is there? And how much endotoxin is present? You can then answer these questions of whether you have enough phage to dose for 4 to 6 weeks, and whether you've gotten rid of enough of endotoxin. The FDA sets the limit for IV administration at 5 endotoxin units per kilogram per hour. So as long as all those numbers work out there, we feel confident in our cocktail. We can take pictures using TEM, and we do that with the UT Health Sciences Center here at Houston. Also, we got all of your typical Myo, Poto, and Cipro viruses. We need to sequence these phage genomes as well, and if we get a large amount of coverage on these genomes, we can be pretty confident in the genomes that are assembled from that data. And Justin is the bioinformaticist of the group, helps us out with that. And we are really looking at that there's no undesirable elements present in these genomes, toxin, antibiotic resistance, and lysogenic elements. So if they aren't there again, then these phages would be considered suitable for use. The other information, if you have this large coverage in sequencing data, is knowing that your phage preps are clonal, that there really should only be one main contig that you are pulling out. If you have more, maybe your preps aren't clonal, or maybe there are some prophages from the host that are coming out. Another important measure that we think about is our treatment plan. So for compassionate use, you aren't going to treat with just phage. You need to also treat with an antibiotic drug. So we want to test at least in culture how that combination, how that pairing would work. And Carmen has developed these assays that we call synograms in lab, where we test increasing concentrations of antibiotic and phage, and of course both in combination. So you can imagine when they aren't present, you get a lot of growth in culture. You can get some killing with phage, and maybe you can get a synergistic killing with phage, which is in fact what we see with one of our best colophages, HB3 here, where we are looking at growth in culture over time, where basically orange means more growth and white means less growth. So at higher concentrations of this antibiotic, you can kill the bacteria, similar with phage, but when they are joined in culture, that killing is greater than either of these just by themselves. We compile all of this information to send to the FDA. We work closely with the doctors to develop a treatment plan, and this process is just a back and forth. Really, we do our due diligence in forming a safe and effective treatment plan, and we just answer and address questions as best as we can in order to approve a phage therapy for compassionate use. If we get to treat the patient, of course, we want to know what happens inside the patient, and what happens after. So we're looking for if we can find phage in blood and urine, and also we try to assess how the patient is responding to treatment also. All this information is gathered and stored so that we can better be prepared for next time, being able to match phages and their pathogens. We have them in digital storage and cold storage, and so an overview of our compassionate use cases to date. You can see, as I mentioned previously, a lot of urinary tract infection, prosthetic infections. Several of these patients have been treated or are being treated, and the good news, there haven't been any issues thus far. The patient's infections have cleared, but of course the real test is what happens after. Have we truly cured these patients and cleared them, or will their infection come back? And if so, is it the same pathogen, or and is it also going to be be susceptible to the phages that we had given them prior? So again, what I had showed you in overview was this entire system that we like to use the term test for our phages: therapeutic, efficacious, safe, and targeted. I went over different ways we can develop and isolate phages, the work involved in the basic science to characterize these phages and develop a cocktail, being able to store this information in the future, and working closely with doctors so we can better understand the basic clinical science of phage therapy. So there's a lot of people to acknowledge here, and again, everyone involved in different labs at the Texas Medical Center. Thank you, and I'll look for some more questions here.
Okay, super great talk, Austen. So there are a couple of questions. Someone wanted to know if you could not find the phages for the Acinetobacter, or you could not find the receptor strain in these phages.
I'm sorry, say that one more time. They were asking about what, trying to find?
If you could not find the phages for the Acinetobacter, or you could not find the receptor strain.
Yeah, so we do have a strain of Acinetobacter for which we are trying to find phages, but we have not found any phages to this point. Again, the basic concept is to go to where those bacteria are. This is a soil. This bacterium is commonly found in soil, so we would be looking at soil samples. But also, again, because it's a multi-drug resistant bug that causes a lot of problems in hospitals. So waste and effluent around hospitals would be good sources to try and look for those phages.
Great. Someone asked, is it possible for a patient to develop an antibody against these phages and have a hypersensitivity reaction?
Yeah. I mean, it is known that patients will develop antibodies to phages. What is not known is whether this is going to be different for different phages, whether some phages are more likely to elicit an immune response than others. We also don't know if dosing plays a role in that process. Also, these are some of the questions we are looking at and testing and trying to answer in the clinical follow-up side of our process, being able to really understand what is going on.
Okay, and one more question. How do you treat patients with a device infection? Do you add the phage to the bloodstream or to the site of infection?
You can definitely do both, and we've worked with both. So if you have access to the site of infection, and say you're scraping some infection out and cleaning it, right? That is a perfect time that when you're finished there to instill phages at at that site. In a lot of these cases, the infection may be difficult to access, and for that reason, that is why we use the IV route to try and ensure that that the phage end up where they need to be. But there's certainly no wrong answer to how to deliver phages at the site of infection. There has just generally been more success, and it's been published using an IV route.
Great. Okay, I think those are all the questions we have for today. I want to thank both of our speakers for giving 2 really great talks today. I also want to thank everyone for attending. We had a really good turnout today, which is always exciting. And last but not least, if you are interested in signing up to give a MicroTalk yourself, Lynn has posted the link to sign up for a talk in the chat. It's at the very beginning, and she just posted it this recently as well. So, okay, everyone, have a great week. Stay safe and healthy.
Thanks, guys.